Dear all, welcome to this video. Please hit the subscribe button so that you automatically get updates for upcoming videos and lectures. Thank you. Enjoy watching.
Now, method of reproduction, as you all know, there are two types of methods, sexual and asexual. The difference between the two is in asexual reproduction, a single organism takes part, there is no need of partner, and this results into genetically identical offspring. Whereas in sexual reproduction, two different partners are involved, that is, two cells are involved, and that's why there is a genetic combination of two bacteria or two cells. Okay, so we will first see the asexual production. They are further subdivided into two types, that is endospore formation and vegetative reproduction. Now what happens in endospore formation? Usually gram-positive bacteria, they release spores. In short, in layman language, we can say that these endospores are the eggs of the bacteria. These spores are released into the environment and these spores further germinate and give rise to another cell. So here you can see this is a bacteria. Now this bacteria synthesizes an endospore. So this endospore is released and the parent cell gets breakdown, and finally after releasing this endospore it will germinate after a certain period of time and it will give rise to a new bacteria. So here you can see that the number of bacteria does not multiply because the original parent cell is going to die. So the number remains as it is, but the only thing is the genetic material will be carried forward for a long period of time. Next is vegetative reproduction. Now in vegetative reproduction there are again three types: binary fission, budding and fragmentation. Now what is binary fission? Binary stands for two and fission stands for division. So a single cell giving rise to two cells is binary fission. So the division of a bacterial cell, first of all, what will happen? Parental cell enlarges and duplicates its DNA. Whatever parental cell is there, it will enlarge and it will form a duplicate copy of the DNA cell. Then a small septum will be formed. Here you can see the septum formation, which will divide the cell into two chambers. And later on, complete cross walls will be formed and the two cells will get separated. So two identical cells will be formed from a single cell. So here you can see that the number of cells are increasing. Okay, so reproduction is giving rise to rise in number of cells. Next is budding. Now in budding what happens is small buds are formed at the end of the bacteria. And genome, so here as you can see there is a starting of formation of bud. Then this genome material will divide and a small copy of it will enter into this bud. And see, now here you can see a complete parent cell has been formed. And in this way, chains of these buds are formed on a single parent cell. So here also again the advantage is what? The number of the cells increases and reproduction results into large population of that particular bacteria. Next is fragmentation. Now usually fragmentation occurs when there are not good conditions or not favorable conditions. So in under unfavorable conditions, what happens is the protoplasm or the cytoplasm, it forms compartments and forms a minute bodies called as gonidia, and these further gonidia give rise to a new bacterial cell. So here you can see the compartmentalization has taken place between the bacteria, and then these individual gonidia will give rise to new cells. So that is fragmentation. Next is the sexual reproduction. Now, as we have already discussed, what is sexual reproduction? In sexual reproduction, two parents are involved, and the offspring has genetic material which is not identical to the parents, but it is a combination of two parents. Now, what are the types? Conjugation, transformation, and transduction. Now, what happens in conjugation is, first of all, do you remember pilus? So, pilus takes part in this conjugation process. So in conjugation, what happens is a genetic material transfer takes place from one cell to another cell. And this genetic material transfer takes place in the form of plasmid, which is also called as F plasmid. The cell having that F plasmid is known as F positive, and the one that is the donor cell. And the other cell who does not have the F plasmid is known as F negative, and it is the recipient cell. Now these are the steps of conjugation. So as I told you, F positive cell has pilus. So this pilus will get attached to the recipient cell. Okay. Then there is an enzyme called as laxosome or transferosome, which will break the DNA into two parts. Okay, it will cut the DNA into two parts, that is the F plasmid, and then it is transferred to the recipient cell. And then after getting transferred into the recipient cell, the recipient cell will synthesize the new F plasmid with the help of DNA polymerase enzyme. So here you can say this is the donor cell which has F plasmid, this is the recipient cell which does not have F plasmid, and the pilus is present on F positive cell. It will attach it. After attachment, this F plasmid small portion will enter into this with the help of relaxosome or transferosome, and finally here with the help of DNA polymerase, this enzyme will get synthesized, and this also will contain an F pilus because F pilus will also divide. So now the old donor cell and the new donor cell are completely identical. Next is transformation. Now, what happens in transformation is the bacteria, say, suppose now there are two cells and the genetic material of the two cells is quite similar, say 90% it is similar. So, in that case, what will happen is, say, A bacteria is dying and it is shedding that DNA material into the environment, and A bacteria genetic material is 90% similar to that of B bacteria. So what the B bacteria do, it can take down that DNA pieces which have been shedded into the environment, and it can get copied or it can get it accumulated or adjusted into its own genetic material. So here you can see this bacteria pink DNA has been shed, and this blue DNA includes this pink DNA into it. So that is transformation. Last process is transduction. Now, in transduction usually takes place in viruses which are infect bacteria. That is the bacteriophage. So here you can see the two conditions may take place. In first condition, say now this is the bacteria and this is your bacteriophage. Okay, so bacteriophage is a virus which infects bacteria. So now you can see that the bacteriophage has purple color genetic material and the bacteria has green color genetic material. So once this bacteriophage enters, what it will do, it will either destroy or it will cut the genetic material of that bacteria into pieces, and here you can see it hit both the genetic material of both the bacteria as well as the virus has been cut down into pieces, and later with the help of other enzymes and other organelles and nutrients and all, new bacteriophages have been synthesized. But whatever new bacteriophages have been synthesized, it may contain the original genetic material, that was the purple color, or it may contain a combination of the bacterial genetic material and the original genetic material. So here you can see that the genetic material, whatever bacteriophages are released, either they will be similar to the original one or there will be slight mutation because due to the addition of the genetic material by the bacteria. In second case, so this may be the first case. In second case, what will happen is, say a bacteriophage, okay, with the mutated genetic material. See, this bacteriophage with the mutated genetic material. Now, if it again infects another bacteria, what it can do, it will transfer this genetic material into the bacteria. So here you can see the green genetic material from the original bacteria has been transferred to the second bacteria. And this green genetic material then is accumulated by the bacteria and a new combination of bacteria has been formed. So indirectly what you are saying is that the original genetic material means the genetic material has been transferred from one bacteria to another through the virus. So here the bacteriophage is acting as a vector and it is transferring this material from one cell to another. So this process is known as transduction. Okay, thank you.